Welcome back everyone. Welcome to 𝗖𝗵𝗵𝗺. We are discussing immunology and this is our fourth session. The topic of discussion for today is complement system. I know that most of you find complements very difficult, but it's my duty to make things easy for you with the help of the tricks, with the help of the mnemonics, and definitely I want these complements to complement your retention capacity of your brain. Okay. Once again, this is Dr. 𝗶𝗵𝗺 and I welcome you to the class. Let's discuss about the complement system. So what is complement system? A complement system is a group of serum proteins that enhances or that complements the ability of the antibodies and phagocytes to clear the pathogens. So basically, complement is complementing the ability of the antibodies as well as the phagocytes to clear the pathogens. Okay, remember all these proteins are heat labile proteins. Coming on to the pathways of complement system. See, there are actually three pathways of complement system. One pathway is classical pathway, other is the lectin pathway, other is the alternate pathway. Okay, simply you can remember with the mnemonic call or so the mnemonic is call, C for complement, A for alternate and L for lectin pathway. Now, classical pathway. Now, my dear friends, let's understand about the classical pathway. So, complements have a very unique thing. Complements activate themselves with one after the other. Okay, so complements activate themselves at one after the other. Okay, so what happens? Why the classical pathway is activated? It is activated whenever there is an antigen-antibody complex which is formed. Okay. So whenever there is an antigen-antibody complex which is getting formed, it leads to the activation of the classical pathway. Okay. Giving you some examples, some important examples, please understand that whenever the IgG or whenever the IgM is going to come, they are going to form an antigen-antibody complex, right? Okay. Now let us understand how this classical system is going to get activated. First of all, the C1 is going to get activated. Okay. I'll try to write this for you. So see the system. So you know the counting, right? 1, 2, 3, 4, 5. All right. Remove this 4 and rewrite the counting. This is 1, 2, 3 and 5. And the 4 is going to come here. Okay. So this is how they are going to get activated. So what is happening? C1 is going to activate C4. All right. C1 is going to activate C4 and it leads to the formation of C4b and C4a. Then the C4b is going to activate C2. It leads to the formation of C2b and C2a. This C2b is further going to, okay, now these C4b and C2b are going to activate C3 and it leads to the formation of C3a and C3b. Now these three, C4b, C2b and C3b are going to activate C5 and it leads to the formation of C5b. Only the B's are important, A's are actually there but not very important as far as you're complementing the immune system. Okay, so basically B's are important for immune system, A's have some other roles. So obviously the C2a, the C3a, the C5a, all these are going to get formed but the major role is being played by them. So I'm revising it for you. What happened? C1 is going to stimulate C4. Okay, and once the C4 is stimulated, it leads to the formation of C4b. Now C4b is going to stimulate C2 and it's going to break down the C2 into C2a and C2b. Now this C4b and C2b combining together in the green color, if you can see, they are going to act on C3 and once they are acting, they're dividing C3 into C3 and C3b. So they are called as C3b convertase. Okay. Now once this C3b is formed, so then C4b, C2b, C3b are going to act on C5 and going to divide C5 into C5a and C5b. That's why these three together are called as C5 convertase. Okay. And finally, once the C5b is formed, this is going to attach with C6, C7, C8, C9 and they are going to attack the pathogen. That's why it is called as membrane attacking complex. Membrane attacking complex. Okay. So for MCQ purpose, what is important? Please remember that your Yeah, that your C4b and C2b are the C3 convertase, yes, and your C4b and C2b and C3b are C5 convertase. So C5 convertase have everything 2, 3, 4, whereas C3 has got 2 and 4 but not 3. Okay, easy to remember. Yeah. Similarly, the lectin pathway, the trigger for activation of lectin pathway is mannose residues on the microbes. So whenever the microbes are coming, they are having some residues, which residues are called as mannose residues, and these residues are going to do what? Going to convert C4 into C2. Okay. So this pathway does not have, so 1 and L look similar, right? So 1, so in lectin pathway there is no 1, directly 4 is going to come and the 4 is going to activate C2, rest everything is same. Okay. And the last but not the least, the important one is the alternative pathway, that is because of the endotoxins, because of the LPS layer, because of the yeast cell wall. Okay, they are going to come alternative pathway. And how it is going to function? So remember the C3b, that's most important. So alternate pathway starting from C3b, that is going to convert factor B into BA and BB with the help of properdin. Okay, it's just you have to remember, please don't put in your brains what is all of that. So just remember that alternate pathway starts with C3b, classical pathway starts with C1, lectin pathway starts with C4, right? And ultimately, finally, they are going to reach to C5b to the C9, that is I told you that's the membrane attacking complex. So very simple, let me just tell you that what is happening. Now this C3b is going to get converted into all these things, and these further are responsible for attacking the C5, and the C5b to C9 is going to get formed. Similarly, lectin pathway C4 to C2 and then C2 to C3, then C3 to C5, and again the same membrane attacking complex, and that is all going to happen. So for MCQ purpose, please remember these, that C3b is the opsonization. So you can remember opsonization as obsonization. So if you can remember by this, you can easily remember that this is going to be C3b. Remember chemotaxis. Chemotaxis is important, and remember chemotaxis. Okay. So yeah. So remember like that. Penta, okay, taxis is also having TA and penta is also having TA. So and A of course is important. So C5 because penta and C5A because it is chemotaxis. So TA is penta 5 and A is there, so 5A. Cell lysis, I have told you, membrane attacking complex C5 to C9. Anaphylatoxin, that means responsible for causing allergy, anaphylaxis. Who all are involved? A, C3A, C4A, C5A. Okay, they are responsible for mast cell degranulation. That means the mast cell is having some granules inside it, and once these granules are going to come out, they are going to release what? They are going to release histamine. Okay, they are going to release what? They are going to do histamine release, right? So this is about the functions of complements. What are the clinical aspects? Remember, okay, let me tell you the sources. So where are these all coming? So remember that C1 is released by the intestine, very easy to remember, 1 and I look very similar. Multiples of 3, 3 ones are 3, 3 twos are 6, 3 threes are 9. 3, 6 and 9 are coming from the liver. Okay, so remaining ones are 2 and 4. 2 and 4 are going to come from the macrophage. So FOUR4 PHAGE. Okay, so macrophage, new spelling. Okay, F and F coming together, 2 and 4. And of course, spleen has got what? Spleen has got 5 and 8 in them. So spleen has got 5 and 8 means what? Remember, double E is coming. So two things you have to remember, that the fifth alphabet of ABCD is, yes, fifth alphabet of ABCD is E. So that's why one is 5 and other E stands for 8. EIGHT. So this is 8. So 5 and 8 are coming from what? They are coming from spleen. Coming on to the last part, please remember, if 1, 2 and 4 are decreased, the condition likely to be is SLE. Okay, very simple, 1, 2, 3 alphabets, and there are 3 complements which are deficient. If your membrane attacking complex is not functioning, that is responsible for recurrent Neisseria infection. Very easy to remember. C5b2C9 is a membrane attacking complex, and please understand that M and N go together. So if MAC is deficient, it leads to what? It leads to Neisseria infection. And then you have got C3 deficiency, that leads to pyogenic infection. Okay, so C3, again you need 3 words, PUS pus, 3 words, C3 deficiency, and in fact CD55 is also responsible for the same, so you can remember with the help of penta. Okay, so penta, that means 55, pyogenic, so recurrent pyogenic infection. And then you have got C1 esterase inhibitor, that is for the hereditary angioedema. So 1 and the first letter of the alphabet is going to be A, so angioneurotic edema, that you have to remember, C1 esterase inhibitor. Alright, so this is it for today's class of complements. Thank you so much. I hope that this video was helpful and now you can retain the complements for longer period of time. Thank you.